Assalamu alaikum students. I am Dr. Asma. Today we are going to resume our parasitology lectures. There had been a temporary break because of the corona situation, but inshallah we will continue with the lectures and finish them in time. Today we are going to start with a new class of the protozoa, which are single-celled organisms, and the class which we are going to study today are the coccidea, and they include Plasmodium and toxoplasma. So basically this is a long lecture, so I've divided it into three parts. Today I'm going to deliver the lecture on the first part of Plasmodium. So just to remind you, the protozoa, they are single-celled organisms and they multiply in the human host. We have been through the amoeba, we have been through the flagellates, and now we are going to start with the coccidea. Important ones are the Plasmodium species, Toxoplasma gondii. They are found in the blood and the tissues. And the other one are the intestinal coccidea, which are found in the intestinal tract, and they are a usual cause of diarrhea. Now about the Plasmodium and their species. There are 4 species of Plasmodium that cause human malaria. Malaria, as you all know, is a very important and prevalent disease in our area and in the tropical areas. So the different species of Plasmodium which cause malaria are the widespread ones are Plasmodium falciparum and Plasmodium vivax, and the lesser widespread species include Plasmodium malariae and Plasmodium ovale. So in our region, in Pakistan and India, the species which are found are the Plasmodium vivax and Plasmodium falciparum. Worldwide, malaria is one of the most common infectious diseases and a leading cause of death. More than 200 million people worldwide have malaria and more than 1 million die of it each year, making it the most common lethal infectious disease. It occurs, as I've already told you, primarily in tropical and subtropical areas, especially in Asia, Africa, Central and South America. Now, let's move on to the transmission and life cycle of the malaria parasites. The malaria parasites are transmitted by the bite of an infected female Anopheles mosquito. So, you all must be knowing that malaria is transmitted by the bite of a mosquito, and that particular mosquito is the Anopheles mosquito, and among these Anopheles mosquitoes only the females are capable of transmitting malaria because they require blood for the maturation of the eggs. So the females have to take a blood meal, and without this blood meal, the maturation of the eggs in the inside the bodies of the female mosquitoes is not going to take place. So when the female mosquito, Anopheles mosquito, takes a blood bite, what it does is it transmits the sporozoites which are contained in its saliva at the site of inoculation into the human host. So this is how The malaria parasite, which is Plasmodium, is transmitted in the form of sporozoites from the mosquito to the human being. Infection can also occur by a transfusion of infected donor blood, by injection through the use of needles and syringes contaminated with infected blood, and very occasionally congenitally when a mother is non-immune. Now let's move on to the life cycle of the malaria parasites. There are two components of the life cycle of the malarial parasite. One is the asexual cycle, which takes place in the human host, and the other one is the sexual cycle, which takes place in the mosquito. So, just to remind you, the definitive host in malaria is going to be the mosquito, because the sexual cycle is taking place in the mosquito. So, just to remind you, a definitive host is the one in which the sexual stages of the life cycle are completed. So, the human host, human beings are the intermediate host, and the mosquito is the definitive host in case of Plasmodium parasite. Asexual reproduction in the human host takes place in the liver cells as well as in the red blood cells. And the cycles of reproduction are called as schizogny. So we have got a hepatic schizogny and a red cell schizogny. And these cycles, they occur repeatedly, and eventually they lead to the formation of the gametocytes. And schizogny is also referred to as mirogony, and a schizont as a mirror. Sexual multiplication, which is also called as sporogamy, occurs in the Anopheline mosquito, in which the male and the female gametes fuse and they form zygotes which develop into oocysts. Each oocyst produces many sporozoites. The sporozoites are found in the salivary glands of the mosquito and are infective for the humans. The vector and definitive host, therefore, as I've already told you, is the female Anopheles mosquito. The sexual cycle is actually initiated in the humans with the formation of the gametocytes within the red blood cells, but it is completed in the mosquitoes with the fusion of male and female gametes, oocyst formation, and production of sporozoites. So, as far as the sexual cycle is concerned, its initiation is within the humans, but completion is within the female Anopheles mosquito. As I've already told you, that the life cycle in the human begins with the introduction of the sporozoites into the blood from the saliva of the biting mosquito. The sporozoites are taken up by the hepatocytes within 30 minutes, and within 5 to 15 days, depending upon the species, they develop into liver schizonts and are referred to as pre-erythrocytic schizonts as well. Pre-erythrocytic schizonts contain many merozoites. Now, what happens is that as the female mosquito bites the humans, it inoculates the sporozoites. They are taken up by the capillaries and they reach the liver cells within 30 minutes. They are found within the hepatocytes. Now, within the hepatocytes, the sporozoites, they repeatedly multiply to form a structure which is called as the schizont, and this structure which is found in the liver is called as the hepatic schizont or the pre-erythrocytic schizont. And inside the schizont, many merozoites are found. Now, one structure or one type of the schizoids which are formed when the sporozoites they enter into the hepatocytes are the hypnozoites. Hypnozoites are the sporozoites of Plasmodium vivax and Plasmodium ovale within the liver cells, which delay their development into pre-erythrocytic schizoids. So, the sporozoites of these two species, when they enter into the liver cells, they go into a state of non-development and they are referred to as hypnozoites. They become dormant forms and they become active later on and develop into the pre-erythrocytones at a later stage, and this is responsible for the relapses which occur with Plasmodium vivax and with Plasmodium ovale. Now, when a hepatic schizont or a pre-erythroschizont is mature, it ruptures from the liver cells, and the structures that it releases within the circulation are the merozoites. So, the structures which are entering into the bloodstream from the hepatic schizonts are the merozoites. Now, these merozoites they are going to develop into the erythrocytic schizonts, and the merozoites they go through different phases of their maturation before they develop into the erythrocytic schizont. And the first stage is the differentiation of the merozoite into a trophozoite. Now, a trophozoite is a ring-shaped structure, and this ring grows into an amoeboid form and then differentiates into a schizont which is filled with merozoites. So, what happens is that the ring form grows, it acquires a portion of the cytoplasm and becomes and this structure then fills up the schizont. Now, when the schizonts are mature, they rupture and they release the merozoites which are present within the schizonts, and these merozoites then infect other erythrocytes. So, this cycle continues at regular intervals, and this is responsible for the spike of fever which occurs with malaria infection. Like this cycle is typical for all the four different species of Plasmodium. For Plasmodium falciparum, this cycle is going to be repeated after every 48 hours, which means that the merozoites which are released from the hepatic cells enter into the red blood cells and they form hepatic schizonts, and when they rupture, the fever spike is going to develop, and this process takes place every 48 hours for Plasmodium falciparum and Plasmodium vivax, and as well as Plasmodium ovale, and it takes 72 hours for Plasmodium malariae. So the fever spike which is very characteristic of malaria, which is accompanied by chills and rigors, is occurring at 48 hours for falciparum, vivax, and ovale, and at 72 hours for Plasmodium malaria. Now, as the trophozoite feeds on the red blood cells. So what happens is the trophozoite is the merozoites, after being transformed in the trophozoites, have to grow. They derive their nutrition from the red blood cell. They eat up the hemoglobin, and what happens is the end product is the production of the end product, which is the malaria pigment, also known as hemozoin. This accumulates in the trophozoite appearing as brown-black granules. When the trophozoite is fully developed, the nucleus begins to divide, followed by division of cytoplasm, resulting in the formation of a schizont containing 8 to 24, depending upon the species, merozoites. As I've already told you, this point I've uh, told you in the previous slide, that the trophozoite, when it is fully mature, the nucleus begins, it undergoes divisions, and every dividing form acquires a portion of the cytoplasm, and this leads to the formation of schizont, which contains different number of merozoites according to the species. The schizont, as it matures, it ruptures from its red cell, releasing merozoites, malaria pigment, and toxins into the plasma, which are the causative factor of the typical malaria attack. Merozoites released from the schizonts enter the blood circulation, and those which are not destroyed by the host immune system infect new red cells, beginning a further cycle of schizogeny with more red cells being destroyed. So what happens is that this cycle of schizogeny and rupture, and then again schizogeny and rupture, this continues on and on. So more and more of the red cells are getting destroyed by these repeated cycles. After several erythrocytic schizogeny cycles, some of the merozoites entering the red cells develop into male and female gametocytes. Obviously, the cycle has to continue, and for the cycle to continue, formation of gametocytes is essential, because for the cycle to continue, the gametocytes have to be taken up by the female Anopheles mosquito, and within the female Anopheles mosquito, the sexual stages of reproduction are going to be completed. So the sexual cycle is initiated within the human beings, but it is completed within the female Anopheles mosquito. As there are some peculiarities about the trophozoites of Plasmodium falciparum, which is cytoadherence and sequestration of Plasmodium falciparum trophozoites. As the Plasmodium falciparum trophozoites mature, antigens form on the surface of the infected red cells, which adhere to receptors or endothelial cells lining capillaries in various organs and tissues of the body. As a result, the falciparum trophozoites complete their development into schizonts in the capillaries and deep tissues, and not in the circulating blood, in contrast to Plasmodium vivax, ovale, and malaria. That's why, in case of falciparum malaria, the most prevalent forms which you are going to find in the peripheral blood smear are the gametocytes. The trophozoites preferably are going to be found within the capillaries of the organs and tissues of the body. So the peripheral blood may not have the trophozoites, and especially it's very rare to find the schizonts of Plasmodium falciparum in the peripheral blood. Now we move on to a few terms which are related with malaria, and one is relapse and the other one is the recrudescence. Relapses are a feature of Vivax and ovale malaria due to delayed development of pre-erythrocytic or hepatic schizonts from hypnozoites in the liver cells. In the beginning, when we were discussing the hepatic schizophrenia, I told you that some of the sporozoites, as they enter into the liver cell, they delay their development into hepatic schizonts and they transform into dormant forms which are called as hypnozoites in the liver cells. Repeated malaria attacks over several years are a feature of Plasmodium malariae also, but these are not relapses caused by dormant hypnozoites, but recrudescence caused by a small number of erythrocytic forms of the parasite persisting in the host. So, relapses are predominantly a feature of Vivax and ovale malaria and are due to delayed development of hepatic schizonts, while recrudescence is a feature of Plasmodium malariae, and these are due to delayed development of erythrocytic forms into erythrocytic schizonts. So these are two different terms. A relapse is going to occur with Vivax and ovale malaria, and recrudescence is primarily going to occur with Plasmodium malariae due to the delayed development of erythrocytic forms. Recrudescences may also occur in falciparum malaria for up to 1 year following the first attack. Now, this is the life cycle of the malarial parasite, and in your written examination, if you are asked to draw the life cycle, this is the simplest form in which you can draw the life cycle of the malaria parasite. So it has got we can say three basic parts. One is transmission. Transmission is to the human host, and then from human host, its transmission goes to the mosquito, and from the mosquito, when it is transmitted again to the human host, the cycle gets completed. So, how this is transmitted to the humans? Now I think you can revise, and you must be following that the transmission is through the bite of the female Anopheles mosquito as it injects the sporozoites into the bloodstream. And the sporozoites, they immediately enter into the hepatic cells within 30 minutes. They multiply by schizogny and they form hepatic schizonts. And some of the sporozoites, as I've already told you, this is a mere repetition. The sporozoites of Vivax and ovale, they can become dormant and hypnozoites in the liver, becoming active later on. Liver schizonts rupture, merozoites are released. These merozoites enter the red blood cells. They then Merozoites transform into the trophozoites. Trophozoites again multiply by schizogony, and a schizont contains, depending upon the species, different from 8 to 24 number of the merozoites, and these then rupture, and again the merozoites are released into the bloodstream. These merozoites then infect other red blood cells, and the cycle keeps on going. And with Plasmodium falciparum, this schizophrenia is going to occur in the deeper capillaries within the body. And during these schizophrenia cycles, some merozoites develop, instead of uh, again infecting the red blood cells and again developing into the schizonts, some of the merozoites they develop into the male and the female gametocytes, and these gametocytes are ingested by the female mosquito as it takes a blood meal, and then there is fusion of male and female gametes, formation of the zygote oocyst, and for formation of the sporozoites in the oocyst, and these are then transmitted to the mouth parts and the salivary glands of the mosquito. And this mosquito is not fully infective, and when it bites the human, the sporozoites are transmitted to the humans, and the life cycle continues. Cytoplasm resulting in the development of a schizont. And this schizont, this is the erythrocytic schizont, it ruptures and releases again the merozoites, which again enter into the red blood cells, and the cycle continues. Now, some of the merozoites, instead of transforming into the trophozoites, they transform into the gametocytes, and these gametocytes are taken up by the male and the female gametocytes. Now, these are the characteristic banana shaped male and female gametocytes of Plasmodium falciparum. The rest of the species, they are going to form circular or elongated gametocytes. Plasmodium vivax, ovale, and malari. Now, these are the sexual forms of the parasite, and they are going to be taken up by the female Anopheles mosquito as it again takes a blood meal, and they are going to enter into the mosquito. They differentiate into a macrogametocyte and a microgametocyte, and a microgametocyte then fertilizes the macrogametocyte, and the structure which is formed is the ookinete, and then are the oocysts, and the oocysts they travel to the salivary glands, and as they rupture they release the sporozoites which are found in the salivary glands, and again from here the cycle continues again. So the infective stage is the sporozoite. So because the mosquito is going to inject the sporozoites into the humans, so the sporozoites are the infective forms, whereas the schizonts, the merozoites, the trophozoites, and the gametocytes, these are the diagnostic stages, and they are found within the humans. So this is important viva question. If examiner asks you what is the infective form of the malaria parasite, your answer is going to be the sporozoid form. And what are the diagnostic forms? These are the schizonts, the trophozoites, the ring forms, and the gametocytes into a mature hepatic schizont, which ruptures and leads to the development of relapses of malaria, which are seen with vivax and ovale species. Now, these are the different morphological forms of the malaria parasite, as you can see under the microscope in thin films, thick and thin films. Primarily, these are the stages which are seen in the thin films. Now, this is the ring form or the trophozoid form. You can see this is Plasmodium vivax, this is Plasmodium malariae, this is Plasmodium falciparum, and this is the Plasmodium ovale. With Plasmodium vivax, the ring form is about 1/3 of the size of the RBC, and it leads to the enlargement of the red blood cell. With Plasmodium malariae, the ring form is going to be thicker. So it is thicker, and it the chromatin appears as a separate rounded structure within the ring, and this gives it a bullseye appearance. These are the trophozoids or the ring forms of Plasmodium falciparum. You can have a double dot. So the different types of ring forms which are found in Plasmodium falciparum. The ring forms can have double dots of chromatin. There may be a single dot of chromatin. There can be multiple trophozoids within a single red blood cell. With other species, you can see a single trophozoid within a red blood cell. But with Plasmodium falciparum, you can see multiple, usually up to 2 trophozoids can be found within a single red blood cells. Then these trophozoids, they can also be found on the periphery of the red blood cells, and these are called as the ecolic forms. And then with Plasmodium ovale, you can see a thick ring form, and there is elongation of the red blood cell, a change in the shape of the red blood cell. That's why it is called as the Plasmodium ovale. Now the rest, you can go through this slide, and then we are going to move on to the next slide. Then those were the developing schizonts, and then we move on to the mature schizonts. So uh, the Plasmodium malariae has got a distinct appearance of the merozoites within the schizont, and this is called as a daisy head appearance. You can appreciate in this slide, this is called a daisy head appearance. And the number of merozoites within a schizont is dependent upon the species. So the largest number of merozoites is going to be found within the Plasmodium vivax, and then Plasmodium falciparum, and malariae and ovale are going to have lesser number of the merozoites, and the pigment, it gets aggregated in the center. Previously, it was distributed throughout the cytoplasm. As the schizont matures, the pigment, it aggregates usually in the center. As you can see, is it has got a typical crescentic shape, and its ends are rounded, and the pigment is distributed throughout the cytoplasm. Fine granules and the which is the chromatin and the pigment, they are scattered throughout the cytoplasm. Now we move on to the now this slide is showing you the female gametocytes of the four Plasmodium species: Plasmodium vivax, Malaria, falciparum, and ovale. You can well appreciate the shape of the Plasmodium falciparum gametocyte. Now, this gametocyte is actually contained within the RBC, but because of its shape, the edges of the RBCs you cannot appreciate them. While in the rest of the three species, you can see the edges of the red blood cells within which the gametocyte is contained, and the female gametocyte is going to be crescentic, and the edges are going to be sharply rounded, whereas the male gametocyte has got broadly rounded ends. And in this, the chromatin is going to be compacted in the center, and the rest of the gametocytes are going to be rounded in shape. Now we are going to stop here, and in the next lecture, which is going to be also going to be on the Plasmodium species, we will talk about the pathogenesis of malaria. Thank you, students. Hope you understand the lecture. Go through it. If you don't understand anything, feel free to put up your questions. I'll be more than happy to answer them. Thank you.